Today we'll be discussing about sex determination as our next topic. Basic idea of sex determination you have got from class 8 onwards. In class 8 you have got, in class 10 you have got sex determination in human beings. So we know that out of the entire set of chromosome, which is called the genome, those chromosomes which control the general characteristics of an organism, those chromosomes are called autosomes. And those chromosomes which controls the gender or the sex of an organism, those are called allosomes. Now in case of human beings, we know that we have 46 chromosomes. Out of this 46 chromosomes, 44 chromosomes, they control the general body characteristics. So 44 chromosomes are autosomes. Whereas the last pair, they are the allosomes because they control the gender or the sex of an organism and associate characters. In case of human beings, now this is the example of human beings that we are taking. In case of human beings, these allosomes, it can be either XX chromosome or it can be either XY chromosome. So that is the concept of autosomes and allosomes. Now when we are talking about sex determination, we are basically concerned about allosomes. So what we have, there are different system of sex determination. The most common one is the XXXY type sex determination. This is in case of female and this is in case of male. Now when this is mainly found in most of the organism. You find them in human beings, you find them in Drosophila and other mammals as well. So what happens here, this is the female, this is the male. So along with the autosomes, which will become half, the females, when they will form eggs by meiosis, they will form either XX, the eggs will be similar, and the male, they will be forming XY. So upon combination, what we have XX, XX, XY, XY. Now what we are trying to say here is that when the males, when they produce sperms, the sperm can be either X sperm or Y sperm, and therefore in case of human beings, the males are called heterogametic and females are homogametic. Why homogametic? Because all the eggs which will be formed, it will be of the same combination, it will have only the X chromosome, whereas the male sperms, it can be an X sperm or a Y sperm. That is one. The next type is XXXO type. Now XXXO, here the female has two allosomes, whereas the male, it has got just one allosome. O means it is missing. There is no corresponding chromosome for this, no homologous chromosome. So when the male will be producing the sperm, one sperm will have X allosome and the other sperm will be blank, no allosome in that particular sperm will be there. So this kind of combination we find in case of grasshopper, cockroach. So these are some examples of XXXO type. This first one, this is the second type. The third type of sex determination that can be seen in case of birds, it is your ZW, ZZ type. The females here are heterogametic because the females, they have Z and W allosome. So from here when eggs will be formed, one egg will have Z chromosome, another will have W chromosome, whereas the males are homogametic. So they will be producing sperms which will have only the Z chromosome. So what we have, Here it is the case of female heterogamety. In case of human beings, it is the case of male heterogamety. So that is the difference. Example of this, we find this in case of birds, reptiles, and certain fishes. The fourth type of sex determination, it is also called the ZOZZ type. In ZOZZ type, O means that in case of, there is also female heterogamety, but the thing is that one egg will have the Z chromosome and the other egg will be blank. Now this type of sex determination, it is found in case of certain moths and butterflies. So these are the four basic types of sex determination. Now apart from the allosome controlling the gender of an organism, there are certain environmental conditions also. In certain organism, it is the environmental conditions which determines the sex. For example, in case of most of the turtles and all species of crocodilians, in their case, what is found that temperature plays a important role in determination of the sex, because it has been found that if the eggs are incubated below 28 degrees Celsius, then most of the eggs, they will hatch as males. If the eggs are incubated above 31 degrees Celsius, then most of the eggs will be born as females. And if the temperature is maintained between this, then equal number of eggs will show. Half of them will develop into female, half of them will develop into male. Another interesting is in case of a worm, which is called Bonellia. It is a marine worm. Now in case of Bonellia, it is found that, suppose this is the larva, it is a marine worm, this is the ocean floor. If this larva falls to the ocean floor, it develops into a large size female. And if the larva, it falls near, the mouth of or the proboscis of the female, suppose the larva has fallen, there is a female, already a female is present, a female is around 13 centimeter long, already a female is present, and the larva falls near the mouth, it enters the mouth and grows as a endoparasite inside the female, then it will develop into a small size worm which is male. So like that, there are different exceptions or other ways of sex determination apart from the normal allosomic sex determination. Now in your course, an interesting case of sex determination of honeybee is mentioned. Now in case of honeybee, what happens? The males, which are also known as the drones, the males are haploid in nature. They have 16 number of chromosomes. And you know that the fertile female is called the queen, which will lay the egg. So queen is the fertile female. The queen is diploid, it has got 32 chromosomes. Now when the males, suppose, okay, we come to the queen first. Suppose the queen, it is producing egg. The eggs will be produced by the process of meiosis. So every egg, the eggs will be having how many chromosomes? 16 chromosomes each. So this is the combination of the egg, 16 chromosomes in all the eggs. Now males directly do not fertilize the eggs. What happens? There is a very interesting phenomenon which happens in honeybee. They go on a vertical flight, which is called the nuptial flight. NUPTIAL, nuptial flight. So the queen flies vertically upwards and all the males or the drones, they will follow the female. The female will keep on attaining height and after attaining the maximum height, only the strongest of the male will go and copulate or mate with the female or the queen. Rest of the males, they come back to the hive. Now, when that male copulate or mate with the queen, the entire sperm sac of the male gets detached and gets stored in the queen, and the male falls to the ground and dies. The queen returns back to the hive with that sperm sac, and after that, during her entire fertility period, she will be fertilizing the egg with the help of the sperms present in that particular sperm sac. And all the sperms in that sperm sac, it is not formed by, there is no meiosis. So all the sperms, they will have 16 chromosomes. Now, if the egg is fertilized by the sperm, then it will form 32, the zygote will have 32 chromosomes, and this will develop, these zygote or eggs, the fertilized eggs, will develop into females. Whereas, if the eggs develop without fertilization, then they will be haploid eggs, and these will develop into males. So this is the kind of sex determination we can find in case of honeybees. Some of the eggs will be fertilized and some of the eggs will develop without fertilization. Just like in case of plants we have parthenocarpy, a similar kind of phenomenon is seen here. So if the eggs are fertilized by the sperm, then it will give rise to female bees. The larvae will develop into female bees. And if the eggs are developing without fertilization, the number of chromosomes will be 16 and the eggs will develop into male bees or drones. So overall, this is the aspect of sex determination given in your NCERT book. In the next video, we will be discussing about mutation and related genetic diseases. Thank you.